Hi, Sushri, is it audible?
Yes, sir, it is audible.
Okay. You can present from there. I'll speak from here. Okay.
Okay, sir, no problem.
Yeah. Did you get the PPT? Is it running?
ରେକର୍ଡିଂ ମାରିବ
ସାର୍ ସେ ଯେଉଁ ଭିଡ଼ିଓଟା ଆସୁଛି ସେଇଟା କେମିତି ବନ୍ଦ ହେବ?
ସେଇଟା ବନ୍ଦ ହେବ ସେ ଷ୍ଟାର୍ଟିଂରୁ ହେବ ତ ତାପରେ ବୁଝିଗଲା ହେବ ଏଇଟା ଆମେ ଅଟୋ ଆଡ଼ମିଟ୍ କରିନାହାନ୍ତି ନା ସେକେଣ୍ଡ ଟାଇମ୍ ପଶିଯିବେ ଆପଣ ଯେତେବେଳେ କଟିଗଲା ଥରେ ନେଇଛି ସେକେଣ୍ଡ ଟାଇମ୍ ଅଟୋମେଟିକ୍ ପଶିଯିବେ ଫାର୍ଷ୍ଟ ଟାଇମ୍ ଆପଣ କରିଦେଉଛି
ନା ବ୍ୟାକ୍ଗ୍ରାଉଣ୍ଡଟା ଚେଞ୍ଜ କେହି କରିଦେଉଛି?
ହେଲା
Sushmit, is it ready?
sir just 1 minute sir.
No problem, sir.
Okay sir. Camera to shall we start sir? Prabhakar sir.
Yes.
Prabhakar sir.
Yes,
Am I audible sir?
you can start now. Yes, I am audible.
Okay sir.
Okay.
A very good afternoon everyone. In the very outset, I seek the blessings of the Almighty to kindly lead us all and bless us to complete this webinar in a successful manner. Respected participants, teachers, students and research scholars, it's an honor and privilege for me to welcome you all to this one-day national level zoology webinar on the topic taxonomy, ecology and evolution of snakes in peninsular India with special reference to the Western Ghats. I also welcome our esteemed principal, Sir Professor Gopal Chandra Behra, whose continuous support and inspiration has motivated us to organize a series of webinars. And for your kind information, this is the third webinar that our college is going to organize today. I also welcome respected HOD Zoology, Dr. Prabhakar Sahu sir, and convener of today's session. He is also Sushriman, co-convener of this webinar. Respected IPSC officer in charge, Professor Satya sir, respected staff members of this college, my colleagues and students. I welcome you all to today's session. Further, it's a matter of great pleasure and pride for me to welcome our honorable chief guest, come chief speaker, Dr. Ashok Kumar Malik. He is a visiting researcher, Evolutionary Ecology Lab, Case LAB, Case Lab, I, on behalf of Udala College and the IQAC and the Department of Zoology, extend my sincere respect and gratitude to you all for giving us time and being a part of this national webinar. Once again, I sincerely welcome you, sir, to this special session. Hope sir's deliberation on the chosen topic will be an enlightening one and immensely benefit everyone present here in this virtual platform. I also thank all the participants once again for their overwhelming support and patience. And as you know, we have more than 500 participants. They have registered for this webinar from different parts of this country and overseas also. So I once again thank them for their support and sincere approach towards us. And with this, I would like to present a virtual lightning of the lamp, and after that we would continue our session.
कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शत्रुबुद्धिविनाशाय दीपज्योतिर्नमोऽस्तुते दीपज्योतिर्नमोऽस्तुते शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः शुभं कुरुत्वं कल्याणम् आरोग्यं धनसम्पदः
Now, I would like to request our Honorable Principal to deliver his presidential address of this session. Over to you, Principal Sir. Principal Sir, over
Thank
to
you,
you.
Panda sir.
Okay, sir.
Thank you, Panda sir. Esteemed resource person, Dr. Ashok Kumar Malik, Professor and Visiting Researcher, Evolutionary Ecological Laboratory, Center for Ecological Sciences, Indian Institute of Science, Bangalore, India. Respected participants, researchers, students of this department, my respected colleagues. In the outset, I thank you all for joining in our third webinar organized by Department of Zoology in collaboration with IQAC, Udala College, Udala. In this respect, I extend my heartfelt thanks to the HOD and convener, Department of Zoology, Dr. P.K. Sahu, and co-convener, Ms. Susisri Sangita Pati, for sincerely organizing this webinar. I also extend my special thanks to IQAC coordinator Dr. Prafulla Kumar Panda sir and assistant IQAC coordinator Satyavartam Malik sir for taking all technical initiatives to upgrade such an webinar in our college with an aim to inspire and update our students, teachers as well as researchers' knowledge during this pandemic situation. It's my pleasure and privilege to deliver a few words about my college. Udala College Udala was established in the year 1965. The institution becomes a full-fledged degree college in 1971. The Utkal University granted affiliation for honors courses of zoology in the year 1988-89. Being one of the premier colleges in the district of Mayurbhanj and in Odisha, as well as we have total 14 numbers of honors subjects. Our students have made us proud by securing top position in the university level every year. This college has accredited with the Gold Plus by NARC in the year 2016. Innovation and rejuvenation have become the catchword to extend the future of this college. Our unity and dedication will bridge the gap between mission and achievements. The topic chosen for today's Ovinus, taxonomy,
Hello, sir.
which shows which shows a high level of species richness, including snakes. Being a charismatic species, snakes always grabs attention from the scientific community and nature lovers due to their high level of endemism and diversity in the and also for actually I congratulate the organizers for arranging such an informative and resourceful session on the very topic. I wish a great success to this program. Thank you all.
Thank you, sir. Thank you for your inspirational words, and definitely such words will motivate us to conduct some more webinars in different departments of this college. Now, I would like to invite HOD Zoology, Dr. Prabhakar Sahu sir, to deliver his keynote address and also afterward introduce our chief speaker of today's session. Prabhakar sir, over to you.
Okay, thank you. And good afternoon to you all. Esteemed Principal, Professor Gopal Chandra Behera, respected Coordinator IQAC Dr. P.K. Panda, respected Chief Speaker Dr. Asad Kumar Malik, respected participants all over India, researchers, respected co-convener of the webinar, Sustri Sangeeta Pausti, Assistant Co-Coordinator of IQAC, Professor Satyabrata Malik, teaching and non-teaching staff of my college, and my dear students. Now I am going to talk about the keynote address of the topic entitled Taxonomy, Ecology, and Evolution of Snakes in Peninsular India, with special reference to the Western Ghats. As we know that Western Ghats, also the Sahedri or the mountain of Asians, it is mountain range covers an area of 1,060,000 square kilometers parallel to the western coast of the Indian peninsula, traveling the state of Tamil Nadu, Kerala, Karnataka, Goa, Maharashtra, and Gujarat. It is an UNESCO World Heritage Site and is one of the 8 hotspots of biological diversity in the world. It is sometimes called the Great Escarpment of India. It contains a very large portion of the country's flora and fauna, many of which are found in India. According to UNESCO, the Western Ghats are older than the Himalayas. They influence Indian monsoon weather patterns by intercepting the rain-laden monsoon winds that sweeps in from southwest during the late summer. The range runs north to south along the western edge of the Deccan Plateau and separates the plateau from a narrow coastal plain called Konkan along the Arabian Sea. A total of 39 areas of the Western Ghats, including national parks, wildlife sanctuaries, and reserve forests, were designated as the World Heritage Sites in the year 2012. The range starts near the Songad town of Gujarat, south of the Tapti River, and runs approximately 1,600 kilometers through the states of Maharashtra, Goa, Karnataka, Kerala, and Tamil Nadu. The area is one of the world's 10 hottest biodiversity hotspots and has over 7,402 species of flowering plants, 1,814 species of non-flowering plants, 1,000 approximate of insects, 149 mammal species, 508 bird species, 179 amphibian species, and 290 freshwater fish species. It is likely that many undiscovered species live in the Western Ghats. At least 325 globally threatened species occur in the Western Ghats. Now, the protection about the biodiversity. The area is ecologically, sensitive to development and was declared as ecological hotspot in the year 1988 through the efforts of ecologist Normal Mayers. The area covers 5% of the Indian land, 27% of the species of higher plants in India are found here, and 1,800 of these are endemic to the region. The range is home to the list of 84 amphibian species, 16 bird species, 7 mammals, and 1,600 flowering plants which are not found elsewhere in the world. The government of India has established many protected areas including 2 biosphere reserves, 13 national parks to restrict human access, several wildlife sanctuaries to protect specific endangered species, and many reserve forests, which are all managed by the forest department of their respective states. The Nilgiri Biosphere Reserve, comprising 5,500 square kilometers of the evergreen forest of Nagarhole and the deciduous forests of Bandipur in Karnataka, adjoining region of the Guyanan Mukherjee in Kerala, and Madumalai National Park, Satyamangalam in Tamil Nadu, which forms the largest contiguous protected area in the Western Ghats. Silent Valley in Kerala is among the last tracts of virgin tropical evergreen forest in India. In August 2021, the Western Ghats Ecology Experts Panel designated the entire Western Ghats as an ecologically sensitive area and assigned 3 levels of ecological sensitivity to its different regions. A panel headed by ecologist Madhav Gadgil was appointed by the Union Ministry of Environment and Forest to assess the biodiversity and environmental issues of the Western Ghats. The Gadgil Committee and its successor, the Kasturi Ragan Committee, recommended suggestions to protect the Western Ghats. The Gadgil report was criticized as being too environmental friendly, and the Kasturi Ragan report was labeled as being anti-environmental. So the 20 different species of snakes found in the Western Ghats of India. As we know that the Western Ghats is the hub for Indian biodiversity and the only remaining rainforest in India. The Western Ghats of India is home of several species of reptiles, insects, mollusks, and amphibians. The major population of snakes species found in the Western Ghats are belong to the family Europeltidae, but Western Ghats is also the habitat to most venomous snakes and non-poisonous snakes. The major population of snake family Europeltidae is restricted to the region. Several endemic reptile genera occur here, including the cane turtle, lizard-like Salia, Respilia, Costlia, snakes like Melanophidium, Plectruros, Peretruros, Platyplectruros, Xylophis, Rhabdops, and likewise. Species level endemism is much higher and is very common to almost all genera present here. Some enigmatic endemic reptiles include venomous snakes such as the striped coral snakes, Malabar pit viper, large-scale pit viper, and Horso pit viper. 20 snake species which are found in Western Ghats are the king cobra, the Malabar pit viper, hump-nosed pit viper, bamboo pit viper, Horso pit viper, large-scaled pit viper, banded coral, green vine snake, multi trinket snake, Wanand eelwag, Travancore wolf snake, Bedom skate snake, Khoiris black siltail, Girish branchback tree snake, Indian red snake, Indian rock python, Indian cobra, Russell's viper, saw-scaled viper, and lastly Indian crate. The topic chosen for today's webinar, taxonomy, ecology and evolution of snakes in peninsular India with a special reference to the Western Ghats. The Western Ghat is one of the biodiversity hotspots in the world, which shows a high level of species richness including snakes. Being a charismatic species, snakes always grabs attention from the scientific community and nature lovers due to their high level of endemism and diversity in the Western Ghats. This talk is related to the snakes found in peninsular India and their ecology. How and why we observe the current pattern of distribution, what evolutionary forces might have shaped their distribution and diversification. That is the aim of our discussion. Now the conclusion of the keynote address. Some of the threatened factors such as overexploitation of natural resources and other anthropogenic activities adversely affect the existing ecosystem, and it may lead to the rarity of many species in future. There is an urgent need for developing pragmatic conservation strategies for the endemic species in the southern Western Ghats, which may lead to their effective protection. So now, before hand over the microphone to our chief speaker for the technical session, let me introduce few lines about him. Dr. Ashok Kumar Malik was completed his PhD degree from Indian Institute of Science, Bangalore, India. He is presently working as a lecturer at Deravis College, Deravis, Odisha. Dr. Malik is a all-time visiting researcher, Evolutionary Research Lab, Center for Ecological and Biological Sciences at Indian Institute of Science, Bangalore, India. Dr. Malik is a hardcore ecologist having passion for snakes. He conducted research in snake evolutionary ecology and biogeographic histories in peninsular India. His dedication and sincere effort in his work finally lead to discovery of 5 new species of Venus snakes. Now Dr. Malik is the section editor of Asian Journal of Conservation of Biology. Okay, thank you all.
Now I would
Thank you, sir. Shall I proceed with the presentation?
sir 1 minute, sir. Just wait 1 minute,
Okay.
sir. Here I would like to request all participants kindly mute yourself because if anyone unmutes, it is a kind of echo that comes out, so others are getting disturbed. So please, I sincerely request all of you, please mute yourself. ଟିକେ ରହନ୍ତୁ ସାର୍। Okay, sir. I am sharing now.
Sir, just a minute, sir. Just a minute. Just a minute.
Okay, sir.
I'll just change the slide, front slide, because it is an old slide, so I just need to change it. I'll share it immediately. Okay, it's okay.
फुल स्क्रीन कर देगी प्रेजेंट ना कले एंटायर स्क्रीन देबे तापर म्यूट कर दो मिनिमाइज कर देगी मोबाइल सुन एटा खुलला परे एटा देखो एटा हाईलाइट कर एटा गोटे क्लिक कर देबे तापर एटा शेयर मार देगी पूरा मिनिमाइज कर दो
Just give me 1 minute.
Have you sent, sir?
Sir, just 1 second. I will send it quickly.
Okay.
Uh, Sorry, it is uploading. Only 2-3 minutes it will take. Sir, send sir. Please
Okay,
download.
Okay, sir. ହ୍ୟାଲୋ ସାର୍ ପଠାଇ ଦେଇଛନ୍ତି ଆପଣ ଡାଉନଲୋଡ଼ କରିକି ସେଇଠି ସେୟାର କରିପାରିବେନି ସାର୍ଙ୍କ ଇଏରେ ହଁ ସାର୍ ଜଏନ୍ କରିଛନ୍ତି ତ ଲାପଟପ୍ରେ ହଁ ଲାପଟପ୍ ଯଦି ଜଏନ୍ ସତ୍ୟ ସାର୍ କରିଦେବେ ସେଇଟା ହେଲା ଲାପଟପ୍ରେ ହେବ ନଚେତ୍ ନେଇକି ଜଲ୍ଦି ପଳେଇ ଆସିବ Ashok sir, please mute yourself.
Sir, is it done?
Just 1 minute, sir.
Sushil, present it with full screen. Yeah, that's nice. Is it audible to everyone?
Okay, sir, fine, sir.
Yes, sir, it's audible, sir.
Okay, so shall I start the presentation?
Yes, yes, yes.
Okay, so good afternoon all. This topic is basically my PhD work, what I have done, and also I'm continuing lots of work currently, um, doing some biogeographic work and ecology and taxonomic work together. So this is a small part of my research work, which I used in my thesis and for the PhD submission and all. So this topic is designed according to the newcomers, those who are young, basically BSc and MSc students, and all the current, the upcoming researchers and all, so that they can get motivated by the talk and they can get some ideas what is exactly going on in the field. So, like one decades, like 1 and 2 decades back, like taxonomy was kind of a dead field. So many of the people, like the youngsters, those who are very new to the science and all, they never like they never like to study taxonomy. They never like ecology. Also, they never like the evolution. But if they know there are lots of things they need to know, and there are interesting things to do, they can study it well. They can do it well. They can do the research in the future. They can study it well. So it all leads to some kind of useful for mankind in the future also. So this is a topic basically which covers taxonomy, ecology, and evolution of snakes in peninsular India. So peninsular India means it's a part of India. This is southern half part of the India, the south half part of the India, starting from Satpura Hill ranges towards the south to the end of our Cape Comorin. Cape Comorin means near the Kerala end, Kerala and Tamil Nadu end. So near the Sri Lanka. So Sri Lanka is not part of peninsular India, but biogeographically, it is a part of peninsular India, South India. It's a part of peninsular India. Okay. So in this topic, I'll talk about the snake. There are many species present in this area, which can be studied in the future, and many interesting questions are there in the biographic histories, in evolution. Also, people are trying to understand their behavior, ecology, and many people are currently doing taxonomic work on snakes, as well as many other taxa like frogs, even monkeys, and many species are there, butterflies and all. So, uh, again, I'm just going to introduce about some parts. Next page, please. Next page. So in this topic, basically, I'll introduce about the Western Ghats. Already, HOD sir already introduced very good introduction to the Western Ghats are, and that is interesting. So people, so these newcomers and the BSc, MSc students should understand what is why Western Ghats is an important area. Okay, and also I'll talk about little bit talk about the peninsular Indian states, and also I'll talk about species endemism. Endemism means how a species is restricted to small area in the Western Ghats and not outside the peninsular India even. Not even they are not found everywhere in peninsular India even if they are just found in small area in the Western Ghats. Okay. Also, I'll talk about the distribution, how they are distributed, and how the diversification process happened using my study in my case study. I'll just show you how the diversification process and everything happened in this area. Okay. So next page. So in introduction, I would like to show you about the Indian terrain. So Indian terrain is variable. If you look at the northern India, North India, it is bounded by the Himalaya. It is rough terrain. It is a long hill ranges. In the western side, it is bounded by Iran and all the places. And if you look at the peninsular Indian subcontinent as a whole, then you can see that we have lots of variable regions. Like western side, we have Thor desert. So it also shows you the different kind of habitat. When you are coming to the central India, you have dry habitat and a little bit of rough terrain in the central India, like Nagpur Plateau and all the Satpura hill ranges and all the places. Okay, and when you are looking at eastern side of India, Bay of Bengal side, you have a long mountain range, but it is discontinuous. That is called Eastern Ghats. Okay, EG. That EG defines Eastern Ghats. And when you look at the western side, Arabian Sea side, then this is a continuous hill ranges, and that is a escarpment. Western Ghats escarpment means it's one side it's very tall, steep, and nobody can climb the western side. But eastern side it is a little bit of leeward side, means it is plain side. So the western side gets lots of rainfall, and it provided lots of because of the rainfall, the forest type and everything is different, and it is very variable. Very variable means geographically terrain and everything variable. And this hill range is 1600 kilometers from Gujarat to the south of Indian part. So it is till the Kerala and Karnataka, Tamil Nadu. So it covers almost 5 states together.
Sir,
Okay.
sir, 1 minute, sir.
Yes, sir.
Sir, actually your
Yeah.
PPT is not showing. Susri ma'am, PPT is not showing.
I can see the PPT. Sir, I can see the PPT.
Okay, sir. I have to pin that one. I am searching.
Yeah. For the participants, it is not visible, sir.
You just pin the second option, sir. Anyone
Okay,
wants
they
to
are
say?
yes, they
You
are
can-
not pinned. Yeah,
You have to pin the second option of the presenter so that you can
Yeah.
see.
They have not pinned. That is the thing. Shall I proceed?
Okay, continue, sir.
Okay, sir. Keep the slide here. So in this slide, you can see the species diversity of snakes. So there are almost 310 species, and more species are coming one by one. Yesterday only one new more species came to existence. A new species described and discovered by the another group of taxonomists from Northeast India. So there are 311 species. I can say 11 or more. We can say more species are there. So I recently described 6 species. So there are 315 or 16 species are there right now, which belong to they all belong to 18 families, means taxonomic families, 18 families, and almost they have 50 genera. So within the 50 genera, there are 310 species. So given this diversity of snakes in the Western Ghats, in India, look at the diversity. You have lots of species. When you compare this diversity to other places, the dry lands and other places, there are very less number of species. But why these species are so many species are there in India, and where are they distributed? Where are they found? How the species richness. Richness is number of species, number of density, species density, number of species present in particular area in India, and where are they present? Go to next slide. Uh, they are mostly present in the biodiversity hotspots in India. So when you look at the hotspots, these are the three hotspots in India where you can see in the map itself. Uh, uh, you can see in the map one of the Himalayan hotspot, which is the Himalaya region, the blue in color is northern India. One hotspot is present in the right side of the map, that is the eastern Himalaya, and that is called Indo-Burma region. Okay, and another hotspot you can see that is the southern India, in peninsular India. So peninsular India is one of the hotspot, and it is a so within the peninsular India, we have Western Ghats, which is a highly species rich area. That is called the Western Ghats section. So in this talk, mostly I will talk about the Western Ghats region. Okay, please go to next page.
Hello, sir. Sir, hello,
So when looking at the Western Ghats, you can see the map in the left side, left bottom corner. So you can see the Western Ghats terrain. This is a continuous terrain, but there is a small gap between them that is called the Palghat gap. In the south part, you can see a Palghat gap. When you look at the terrain in there is a terrain image below. Uh,
sir,
Look at the terrain image.
sir,
Uh,
sir,
some
sir,
yes.
just one thing, sir. Just hello, participants. If you want, if your PPT is not showing in your laptop or mobile, you just pin the presenter, and that will show you. Pin on the presenter.
Please, sir, please mute some of the participants because somebody is
Okay,
using mobile outside and he
okay,
does work.
okay, sir. Okay, sir.
Otherwise, just remove him from the presentation. Just remove him from the presentation. NK Babuli, just remove him from the presentation.
Okay, sir.
Kindly remove him from the presentation or mute him completely. Hello, sir, is it audible?
Okay, sir. Fine. Fine, sir.
Yeah, I removed that person. So, uh, so the Western Ghats terrain. When you look at the Western Ghats terrain, there is a gap. You can see the red arrow in the right side corner. That gap is very low. While the hill ranges are very high, you can see the left side one is the Nilgiri hill ranges, and the right side one with Annamalai and Palni hill ranges. Those are the 2,500 altitude from the sea level. So that is the only gap which provides interesting pattern to the Western Ghats. So basically Western Ghats, sorry. So basically the Western Ghats terrain and everything is so variable, you can find different kind of habitat type and different kind of forest from the north to south. So towards the south of the Western Ghats, you can get more dense forest and more rich rainforest. It's a very humid and rich rainforest where you get most of the species density in this region. Okay, so this is the region, and in the places like other parts of the Western Ghats, you have high altitude grasslands where you can see the grassland as well as woody forest together. So those grasslands are very important part of the speciation process. So they form the species evolve there on those grasslands, and you will get the woodlands. Woodlands means that those are the rainforest within the grasslands, and those it is so humid inside the rainforest. It provides so much opportunity to the species, and it provides so much competition among the creates so much competition among the species, the species to within this humid habitat. And so from Western Ghats, from the north to south, you will get species turnover. So including any kind of species, including snakes, frogs, plants, anything. You get a species richness towards the south. When you walk from Maharashtra towards Karnataka, then towards the Tamil Nadu and Kerala, you'll get more and more species, and those are more endemic to the southern part. And So most of the endemic species, they are restricted to the most southern part, including this Nilgiri hill ranges and all the central western Ghats in the central region, as well as in the Anamalai and Panlin, Debarmalai, and all the places in the southern western Ghats. Next place, next page, please. So all the distribution pattern, when you look at the species turnover across different area, different species found, different snake found in different area, and uh, and their similarity is equivalent to the some species found in the Malayan region, Malaysia and all the region, Malaysia, Indonesia, Thailand, all the Southeast Asian countries. So basically, this is called the Oriental region. Basically, all together, including India, this is called the Oriental realm. When the students are studying the biogeographic geographic regions, they must be knowing what is the Oriental region. Plus, the student must be knowing, all the PG student must be knowing this thing, Oriental region. So this is within the Oriental region, India is situated. So, so most of the species show the similarity with the Malayan species, Thailand species, or the most of the species found in this Java and Sumatra and Borneo and all that. But why? Who never nobody studies it ever. Why they are similar? And many species which are present in the Western Ghats are not found in the central India or not even in the Eastern Ghats. Only one or two species can be found in Eastern Ghats section, uh, but more species, more five, seven, eight something species are found in the Western Ghats. When you are looking at the frogs or any other species, there are more species in the Western Ghats. Like there is a bush frog, Pilatus or Austristis, which is like present in the Western Ghats. Lots of species are there, almost 50, 60 species are there. But in Eastern Ghats section, only one or two species are there. So this is what the richness which provide the Western Ghats provide, but why and how. So go to next page. Uh, Next page. So there is a old hypothesis people described earlier. It's very old hypothesis. This is called Gondwana land movement, plate tectonic movement. Okay. So what happened in the Gondwana land and plate tectonic movement? The India was a part of a large landmass called the Pangaea. So this Pangaea includes South Africa, Africa, all the places, Africa, America, and all the Australia together. And around 250 billion years back, they got separated from each other. And India got separated. You can see the image video in the top of the slide, how India is moving from Madagascar and Africa and going towards the north. And it is colliding with the Eurasian landmass, which is Asia today. Eurasian landmass is China and Russia. So it is got colliding. So that, and after that, when it is traveling for last 60 million years, it developed lots of fauna species, which speciation event happened there. Lots of species created, lots of species got extincted for last 60 million years. Then it collided with the Asia and the Eurasian part around 30 to 40 billion years back. After collision, you can see the Himalayan landmass formed. So before that, when just after the collision, lots of species colonized to the different area. Some species came into India, some species come out of India, some species come from the Eurasia, mainly from Ethiopian region, African region, also from Malayan region, many places. But after completion of all the collision, all the formation of all the landmass, landmasses like Himalaya region, and also the Thor Desert, which cut off the species colonization to the Indian part, towards the southern India, so towards the peninsular India. So till the recent time, only one route is available, that is the Malayan route, through where Malayan fauna came to India, Indian fauna came to India. So this is a hypothesis. This is a hypothesis proposed long back. So it needs to be evaluated by the people. Okay, through the different taxa. Let's go to next slide. So there are like many questions arise from all the facts we can see from the all the background information, that how many species are present there, whether there are lots, numerous more species are present there, and why and how the endemism, why some species are restricted to the Western Ghats only. They are not found in Central India, they are not found in the Eastern Ghats, but some of their sister species are found in northeast India, like Assam, Meghalaya, Manipur, Mizoram, as well as Myanmar and Thailand and all the places. Also in some parts of eastern Himalaya and western Himalaya. Okay, and why some this pattern, this species distribution pattern is so much discontinuous? Why the species are discontinuous pattern? There is a gap where patchy distribution. Some species are distributed patchily. Some found in Assam, some found in Western Ghats. They are not found in central India. Some one species found in the Eastern Ghats. Some species found in the Vindhya Himachala region, but not everywhere. Why? This why? So this is another question. So second question, the last question I asked mostly the question is the species radiation and in situ diversification. What is a species radiation? Species radiation is basically from a single ancestral species, two species will form, three species will form. So basically, it's a doubling of species from one species to two species, four species, eight species, and number of species. And within this process of species radiation, what happened? Uh, some species will go extinct. So that the radiation process will happen with extinction as well as speciation event. And so this speciation and radiation event also known as the in situ diversification. Means from a single location, species will be diversified from a single species to multiple species. So whether Western Ghat is part of this in situ diversification and species radiation, that is another question. So please come to the next slide. Somebody is talking. Please kindly switch off your mic. Most of the time when you go to the Western Ghats section, because it is the variable environment, variable ecology, with variable climatic factors, variable forest size, grasslands, including your hilltop grasslands, scrublands, many type of and rainforest, semi-evergreen, dry deciduous, moist deciduous. Dry is not mostly, dry is the northern side mostly. So moist deciduous forest and all that is together, it provides opportunity to species diversification in this Western Ghats section. So these pictures mostly taken by me, and these are the endemic species found in the Western Ghats. Okay, so most of the vipers, many of the colubrid snakes, they cover almost 60% of snakes in the world. They are non-venomous snakes, and they are the good runners. So they are the colubrid snakes, and many species of snakes are found in the Western Ghats. Okay, go to next slide. So just actually I gave some introduction about snakes and all. So out of those, I told about the 18 families are present in the India. In India, so you have out of 18 families, 8 families who have endemic species in the Western Ghats. Okay, so 8 families they have endemic species in the Western Ghats itself. So out of those species, I selected some of the good group of number of species to understand all the questions I asked for. So there are there are some boas belong to the python family. There are some wood snakes. You can see the wood snakes. They are endemic to the Western Ghats. This is the genus. This is a genus. Basically, the xylophids. They are endemic to the Western Ghats itself. And there are cyclops, blind snakes. Many blind snakes are there. There are coral snakes. Coral snakes are some coral snakes are found in the northeast region. Some of Thailand, Malaysia, Mizoram, Manipur, and all the places, Southeast Asian places. And some of the coral snakes only found in this section, basically the Western Ghats section and Sri Lankan section. Some 4 or 5 species are present there. Okay, so there are lots of naturish insect snakes like keelbacks, our water snake, checker keelback, bhondo sapo. So checker keelback and all the different endemic species are present in the Western Ghats. Also, there are vipers. There is a very common viper called Malabar pit viper. I'll come to that topic next time. Next slide. This Malabar pit viper widely distributed all over the Western Ghats, and there are like 4 or 5 species of pit vipers present in the Western Ghats, including our common viper like Russell's viper and Soskel viper. All they are present in the Western Ghats. So there are more endemic pit vipers are present in the Western Ghats. Also, there are I told about the colubrid snake. There are colubrid snake covers almost 60 to 70% of the snake population in the world, population and diversity. So there are vine snakes, there are cat-eyed snakes, there are bronze bat snakes, there are rat snakes, There are many snakes, but every group, every genera within this colubrid have at least one endemic species found in the Western Ghats. Most of them have a lot of endemic species. They are just restricted to the Western Ghats. They are not found in Odisha also. They are not found in everywhere in the peninsular India. They are not found in all over the India. But some of their sisters, you can find them in the uh, northeast India, basically the Mizoram, Manipur, and the Assam and Arunachal Pradesh and all that side of. Also, Sir told about the European snakes, those are called earth snakes, which are completely restricted to the Western Ghats and Sri Lanka. There are almost 40-50 species. Currently, people are discovering new species out of this group. And this is a very interesting group. Uh, they have a shield in their tail, and these are mostly boring snakes. They survive under the burrows and under the ground or subterranean species, terrestrial species mostly. Next slide. Next slide. So there are lots of species. I just work on some of the 7-8 genera, different group in the Western Ghats and Peninsular India. Out of those, I would like to present only 2 or 3 genera because it will take lots of time otherwise. So this is one of the group called vine snakes. They belong to the Columbidae family, and they are known as the Ahatula genus. They belong to Ahatula genus, mostly arboreal snakes, and they are found in all over the Southeast Asia. You can see the map where you can see all the black triangle everywhere. Those are the locations of each and every holotype. First description is each and every species found in those areas. Okay. So Odisha also have at least 2 species found in Odisha region. Also 3 species are there in Odisha technically now. So there are, when you look at the red, blue, green dots in the left side of the map, Indian map, the Western Ghats map, you can see actually the Western Ghats have the number of species found in the Western Ghats. Those are the species recently I described. I will come to the next section. So these are arboreal snakes. They are non-venomous snakes, mostly found on trees and bushes. And some of the species are so specialized, they are found in grasslands in the Western Ghats section, in the high altitude areas. And some species are found in the lowland. Next slide. So these are 2 species of vine snakes which are found only in the Western Ghats. So they are known as endemic species. So they are separated by a gap. I told about the gap in the Western Ghats, that is called a Palghat gap. So this is 24 km wide for gap between the hill ranges. So it separated them and they spaced from each other in their morphology. You can see the presence and absence of some character in the picture being down between the 2 species. And they are highly specialized species in the grassland, and they survive those grasslands. Basically, in night, the temperature falls to 0 in the Western Ghats. So during monsoon and during the winter, this temperature is so low, and daytime the temperature is so high on those grasslands. The species, how they accommodate in that temperature, that is very interesting. That is the ecology of these snakes. They survive in the top, and when you bring these snakes to the down, they will ultimately die towards the altitude almost 2000 plus meters, 2000 meters above the sea level. When you bring them towards the 700 or 500 meters, they will die because of the ecological variation. They cannot survive in the ground level. So these are the 2 species found. So there are many species like this. They are just restricted to a small areas in the Western Ghats, and they are the endemic species, and they are not found in the anywhere in the peninsular India, not or in the all over the world, or all over the Southeast Asia and all over the Asia. But their sisters are present in the lowland. Only 1-2 sisters, and some sisters are present in Southeast Asian countries. Okay. Next page. So these are 2 sisters. Another 2 sister species, basically the brown vine snake, which was considered to be the species continuously found in the Western Ghats, as well as in the Sri Lanka. But recently, this species is separated from the Sri Lankan species. So Sri Lankan species and this species are separate. And the common green vine snake, which is also present in all over the peninsular India. which is overlapping everyone's habitat. Basically, it's a sympatric species. It's overlapping everyone's habitat. So it's a sympatric species. It's found in Odisha. It's found in the central India. It's found everywhere till the northern India and all the places. Next slide, please. So another group I am interested, that is the feed viper group. So this feed vipers are called the Tridosos or Craspetocephalus group. They have a pit. Basically, they have a pit to sense the environment and sense the chemical and temperature and everything. In below their nose, just behind their nose, there is a pit in the feed viper. So this species you can see here. It also found in Odisha. So this species can be found in Odisha. This is a bamboo pit viper, which is normally found in all the Mayurbhanj district and all the places from all over Odisha, from all over eastern Ghats, and mostly in the western Ghats also. Also, this species is the first species which was described scientifically around 1933 or something by colonial British rule time. So they described this species, and after that, slowly, they described loads of bamboo pit viper, and they separated the species from this species. So go to next slide. Next. So you can see the map right now, where you can see the dots in the map. So from a single species, there are recent days there are 51 species found in this group. So in pit viper group, in Trimeresurus and Craspetocephalus group, you can find 51 plus species. Means more species are coming to science. Okay. Out of this 51 species, 4 species are found in the peninsular India itself, and one species which is found in Sri Lanka only. So there are, you can see the central Indian part and the peninsular Indian part. Only one species in the peninsular India, which is distributed uniformly everywhere in the peninsular India, but 4 species. Out of these 4 species, 3 species are restricted to the Sri Lanka, Western Ghats itself. Why? And they are just like the fire snake I just described few minutes back. They are just restricted to a small distribution range. Okay. So these are the 2 groups I widely studied, and I'm studying on them. So next slide, please. So these are the endemic species found in Western Ghats out of this pit viper. When you look at the large scale pit viper, it's a morphologically very unique pit viper. It has a large scale, very large scales they have in their body, mostly found in tea gardens. It is endemic species, and mostly tea gardeners get bitten by the farmers get bitten by this snake. But they are mildly venomous. They are not mildly, they are not that much the poison, that much not that much potent. It cannot kill a person, healthy person. So it can give you some symptoms and some pain and all. So this is one of the species. And when you look at the Malabar pit viper, Trimeresurus malabar, the left side species, this species is found in all over the Western Ghats. Okay. And this Trigatos is a restricted species. It's found only in the Nilgiri Hills. Okay. So one species is found in Trigatos is found in Nilgiri Hills, and Macrolepis, this Trimeresurus macrolepis, the green one, is found in the Anamalai Hills. So they are separated by the Palghat Gap. Okay, and they have a small restricted area, and the habitat temperature climate is very hot there, and in the grassland habitat they are specialized species mostly, very specialized to their ecology, and they are restricted to a small area. If something happened tomorrow to those grasslands, because those areas are very small, and maybe the climate change or something will impact on them, then definitely these 2 species are going to extinct very quickly, because they don't have a large range size. They are restricted to a small area. And the 4th species, the Trimeresurus gramineus, which is bamboo pit viper, it is found everywhere in India, including the Western Ghats. So it is in central India, it is in northern India, it is not in Himalayas. Himalaya, another group of pit vipers are there. So this is another group which is present in all over India. So this is the second group I worked on. So there are a few groups I am working on. I'll talk about at the end. So go to next slide. So before proceeding to this talk, why I need to understand the species and number of species, because when before proceeding to any of the ecological work, or maybe some for the conservation perspective, when you are giving priority to some area to conserve, you need to count the species, how many species are present. So basically, species is a basic unit for conservation. So you calculate the unit number of value and you conserve those areas. So that is the first question. And second question, how many gene pools you can conserve from the conserving a species. So when you are conserving a species, you conserve a gene pool itself. And third question is, when you are in the future, when you are going to study something like behavior or biogeography or ecology, history and everything, how the species evolved and everything, you need to understand each and every lineage. You need to separate the lineages. You need to separate the species from one to from separate from each other, so that you can draw a line and separate the species, and you can make your decision, and you can do the behavior work, you can do the ecological work, you can do the biogeographic work. So I did the first thing I did is the to understand the species boundary. So I what I did to understand that, I sampled across India. I sampled across India for many species from the each genus. So these are the some sampling locations in the star mark you can see in uh, in the map itself. So Indian map itself. These are the sampling locations. And second thing, I collected the morphological data and specimen also I collected from the regions. Also, I collected tissue samples for their DNA analysis. Also, I collected GPS data location. What is GPS data location? This is basically, most of the mobile holders nowadays knows what is GPS. So GPS is a global positioning system. Where are you standing? If you have a positioning system, you have a coordinate from for that area. But GPS device provides more information, altitude, latitude, longitude, and all the location. Many information it provides from that terrain basically from particular region. Also, for this study, I sequenced and used the mitochondrial nuclear DNA, basically 5 genes from cytochrome B, ND4, 16S, 16S, and RAG1, CMOS, all the nuclear genes. This set of genes are used sometimes because mitochondrial and nuclear genes are used because uh, these mitochondrial genes are very fast evolving genes. They evolve very fast. Okay, they can provide you a depth between two species. So what is the distance between two species easily, and they can provide you how when they evolve from each other very quickly. And the RAG1 and CMOS, those are the nuclear genes. Basically, it provides a deep depth. Basically, they are slow evolving genes. So in a thousand base pair of the sequence, you can get only 5 to 10 mutation or 15 mutation in the base pairs. Basically the purine and pyrimidine base pair mutation. Basically, those are called as substitution or mutation. Basically, so but in mitochondrial gene, you get a large number of mutations, maybe 150 or 250, 200 mutations in 1000 base pair, basically. So in the end and the bottom below, I'm just giving you an idea that how a DNA sequence looks like. Okay. So these are the sequences I sequenced, the 4, 5, 6 I sequenced, and 1 to 2 dozen sequence I acquired from the gene bank. Okay. So these are the DNA sequences for cytochrome B basically. So cytochrome B, when you look at the sequences carefully, you can see there is a difference in base pair. Sometimes A replaces, sometimes C replaces, sometimes G replaces. Those are the mutations, and those mutations can be found in two different species. From species to species, the mutation varies, and number of mutation varies from species to species. So it will give you an idea when you comparing to sequence from species A to B to C. They are very different mutation. They are we have species A, B, C. Okay. Go to next slide. So how the delimitation process I did for the snake, mostly this is the approach mostly people use these days. This is the approach we basically developed from IIC, and all over India and all over the world people are using this strategy to delineate species. Means they separate species, discover species in this way. So this is the way how to describe the species. Basically we construct a phylogenetic tree. So phylogenetic tree is based on the DNA sequences we sequenced just a few minutes back. I told about that. So the phylogenetic tree will give you a different. Will give you a. It will derive it. It will provide you output of the tree on the basis of their mutation. You can see the top branches. Many of the students, they must be knowing what is phylogenetic tree, and all the teachers, they must be knowing all the phylogenetic tree. So this is the phylogenetic tree where species to species correlate from this common ancestor. Okay. So when you talk about the branch length in a phylogenetic tree, it is on the basis of substitution, mutation. Okay. So this is the process where the branch length, the length is derived from the number of mutation and substitutions. Okay. So basically, this is the phylogenetic tree we derived from maximum likelihood phylogenetic tree for a group. Then what we did for this species delimitation to separate species, we used a method called the Bayesian position tree processes. This is an algorithm which calculates we uses the branch length and the topology means the tree topology. The topology means the phylogenetic tree topology. It takes both together and it provides you some clustering, means some independent lineages, so evolutionary lineages. So basically, each lineage is not a species. Okay, it will provide you as a species, but you have to reconsider again to understand whether it is a separate species or not. So it will provide you an output tree showing that it is species. So the right side bottom image you can see in a phylogenetic tree. Basically, one image is this is the tip in the tree is one. The top one is called tip, and when you take a cluster together, that is called the head. Okay, so in a clade, there can be number of species, there can be individuals of a single species, there can be genus, several genus, there can be several family within this group. Okay, so this is the delimitation method. So after taking this next slide, so after doing that, how you delimited the species type species again? So we use multi-criteria approach to delimit the species. So basically, in the multi-criteria approach, we use the morphology as well as geography from the BPTP, because it only BPTP provides you only lineages, putative lineages. Those are not truly species. You have to look at them as different species. So basically, when you look at them as through the morphology and genetic distance, when the genetic distance between species A to species B is high, we kept a cutoff value and we made them a separate species. Okay, and when it is low, then we always consider their morphology and geographic separation. So geographic separation means there should be some gap. So two species are distributed two sides of the river. Suppose Amazon river, two species are distributed from one bank, another bank. So separated by the Amazon river. So because of the Amazon river, those species specified, they never meet each other and the species become separated. That is a gap. That is called the geographic barrier also. So in Western Ghats, we have a gap called the Palghat gap, which is an important gap. So in the map, if you look at the stars, those are the two species present in the Western Ghats section. So they are separated by a geographic gap. Okay. So this is how we use morphological data, we use genetical data, we use other putative methods, we use geographical data, and we separate the species. So how many species we found and how species found, I'll show you in the next slide. Okay, come to next slide. Okay, so this is the cutoff value I told about. The BPTP high divergence, there is a new species, and the BPTP low divergence, then we consider the morphology and geographic separation, and we describe them as separate species. Okay, come to next slide. So in vine snakes, if you look at the result, most of the people, these are the phylogenetic tree. You can see both sides. These are the trees where species are separated from each other. When you look at the right side tree, so this is a paper published recently from my work. So I described 5 species. Also, I found 2 species which were described, documented earlier, but never considered there. So in this picture, you can see that there are lots of species present. They look similar to each other almost. So that is the reason they considered to be a single species earlier in the Western Ghats. But when I looked at the geographic separation or morphological separation, they become the different species. In the left side of the map, this slide, if you look at the left side, the green region, mostly the green region, green color region is peninsular Indian species. Okay, and when you look at the blue region, blue color region in the picture, uh, that is more those species. Those are the Asian and Sundaland species. Sundaland means Southeast Asian species. Okay, Asian species. So peninsular Indian species, there are lots of species now. So if you look at the number of putative species, putative means they are not certain species. I found 26 species within this group, including all the existing species. Okay, but in peninsular India, I found 16 species. But truly, how many species are present in peninsular India and there? Only 10 species. Means 6 species are new to the peninsular India. Okay. Then Sri Lanka has 2 species and Sundaland has 9 species. So in genetic distance DEG, you can see the left side DEG, that is the genetic distance wise. I separated 20 species, and morphologically wise, I separated 14 species. But geographically wise, I found more species because morphology is deceptive. Morphology will never give you the answer immediately because many species are cryptic species. They are called the cryptic species. You cannot diagnose one species from another. They look similar, but when you look at the genetics, inside the genetics, you will get more species. The genetic divergence, genetic restructuring will be there. They will be separated by genetic. Okay, they are isolated by genetic, isolated by geography, but morphological they are not isolated. Morphological they are similar. They are the cryptic species. So these cryptic species you need to approach different way to separate them. That is a geographic and genetic part. So you always use the geography and genetic part to separate these two species, these cryptic species basically. So these are the number of species I described, I found in the Western Ghats. So many new species like total species it is now 17 species in India. So all over the world there are 70 species in vine snakes. On the basis of it, there are more species present actually. Next slide please. So when you look at the map itself for the vine snakes, all the vine snakes, all the dots, colorful dots, show you different species found in different regions. They are kind of sympatric. Sympatric means they are overlapping each other. Some species are widely distributed. Some species are restricted by geographic barrier. So they are mostly geographically restricted and morphologically similar species, but genetically they are also restricted. Okay. So there are some other factors which separate them as different species. Okay. Go to the next slide. So in the process of this taxonomic studies also we found a very ancient lineage which was never described or never informed earlier, never discovered earlier. For last several decades, many decades, Britishers and many archaeologists worked in Western Ghats, but they never found a species which is very deeply diverse lineage, and we never got any museum specimen also. So this is the another species. We named it as Pro Ahatula because we separated it as a separate genus on the basis of their morphological characteristics. And this is one of the ancient genus. That's why we named it as Pro Ahatula. This genus is basically 26 million years separate. It is separated from the Ahatula, this vine snake, from around 26 million years back. So the common ancestor was there, and from the common ancestor, both the species separated, and the morphology and everything changed around 26 million years. That is the Oligocene period, mostly the early Oligocene and all. Okay. So this is the time period. You can look at the tree, and below you can see the 26 million years. That red arrow shows you the separation between these two species, and these species are lower. Interesting scale pattern. They have a rock scale. They have larger number of scale. They have lots of post-ocular scale on the head. And so the dentition, the maxillary, we found the maxillary numbers are more in the species. So this is the species we found in the process of this taxonomic study. Uh, Go to next slide. So, sorry, I just explained about the primates, speech vipers, how they are distributed. So similarly, we did for the primates, all the methods and everything approach we used, and we found uh, eight morphological species and geographically 10 species. So when you separate them by geography, you found the 10 species. When you separate by morphology, you found the 8 species. What happened with the two species is not in the morphology because they are cryptic species. In cryptic species, you cannot separate them through the morphology, but you can separate them through the genetic data. So each bar in the tree, you can see the left side of the tree, the green regions, mostly the green regions are from the Western Ghats, and blue region is from the Northeast India, okay, Southeast Asia. Uh, so this is a peninsular Indian species, all Western Ghats and Sri Lankan species also there in the green region. Okay, so when you look at the bar on the right side of the tree, each bar indicates a different analysis. Basically, the P3, which is a blue in color, and the gaps, so that each species. So in the P3, if you count 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, then there are 12 species according to the Bayesian process analysis. Okay, but when you look at the genetic distance, you got little less number of species. In morphology, you get more and lower number of species. In geography, you get little bit more number of species. So this is how we approach this group and separate this group. Okay. So what we do, why we are doing this? Because we need to understand how many species are present there and how many lineages are present there to understand the further biogeographic analysis and to carry out to understand their geographical distribution pattern and everything, diversification pattern and everything. Okay, go to next slide. So we know now how many species are present and how many species are we separated them. So now we are going to another section which is called the historical biogeography and species identification. This is to understand whether Western Ghats played an important role as a in situ radiation, whether species originated from Western Ghats, whether species diverged from Western Ghats, whether species speciation event, formation of species happened in Western or is it happening in the Western Ghats? Yes, let's see and let's go to the next section. Okay, next slide. Next slide. Go on. Yeah, stop there. When you look at this section, how I have to approach, how I have to understand. So there are certain methods I use. Mostly there is a method called the BEAST. BEAST it will provide you the chronogram. Chronogram means time period of a species when the species is evolved basically. So this is a molecular clock. You know the molecular clock, molecular dating. We date the species and we say that dinosaur belongs to 65 million years back. How they know? This is because of the molecular dating. Okay, so this is a process where we use the DNA sequences for the molecular dating, and also we use the calibration. So BEAST is one of the computer programming, which will use the use your molecular marker genes that DNA sequences you acquire, and also use the fossil calibration. Fossil calibration means I use the 6 fossil calibration for this group. Okay, 6 fossil calibration means some fossil found some 5 million years back from Sri Lanka, suppose. So I use that species and compare that fossil to the one of my species, and if it is similar, I can consider it as the fossil for that group, extinct fossil. So I can use the 5 million year gap in the tree analysis. So it will provide you a molecular clock. It will provide you a chronograph, a dated tree with how many million of years each plate separated. So it will provide a evolutionary age for each species from their ancestor. Okay. So second thing, what I did is that biogeographic reconstruction, so that I can understand how they are distributed and why they are distributed like this, why endemic species are present there. Okay. So basically, in this reconstruction area, I separated all the parts of the Asian parts to the different section. Basically, the peninsular Indian as one of group, and where I separated peninsular India into two groups. One is the Western Ghat, and another is the drier parts of peninsular India. Second, I separated Sri Lanka because Sri Lanka is separated landmass from Indian landmass, and it has spaces similar to Indian Western Ghat. Okay. And so Asia and Switzerland. Asia is mostly in the China region and all the places, and Sundaland in Southeast Asia, mostly Thailand, Borneo, Philippines, and all the places. So that's a Sundaland. So I use this five areas as separate area. And next, what I did three major analysis to understand understand their distribution and ancestral area. So basically, one analysis is called BBM. So what it does, it provides you an ancestral area. Means whether the species ancestor was present in that area, particular area. Suppose whether this analysis will provide you the area. Basically, if it is present in peninsular India, it will give you a value, some probability value, so that you can know that the species was present in the peninsular India, but now it is not present there, but it is present in the Western Ghat. Means this peninsular India is one of the part which may be participated in the speciation process. It may be gave rise to the new species from there. Okay, but the species is now present in the Western Ghat, kind of like that. And another analysis is called the Lagrange and DEC. So DEC is this area will give you the ancestral species range. Okay, means whether this species was present somewhere else, and it came to Western Ghat, and it evolved there. But it will also show the species come from somewhere else. Suppose it is present in Asia, Sudaland or somewhere else, and it came to Western Ghat, then it will show that the Western Ghat was Sudaland was one of the ancestral range for this group, for this species. Another analysis I use, that is the SDBA. You can see the SDBA. So that is called statistical dispersal and vicariance. Okay. So dispersal means one species dispersed from India to Sri Lanka, suppose. So India and Sri Lanka are separated. So one species dispersed means it can swim and go to Sri Lanka, and in there, what it will happen, it will give rise to different species. Okay. Suppose one monkey will go from Sri Lanka, from India to Sri Lanka, and there that species become different species, new species there. Okay. So that is called the dispersal. When but what happened? The vicariance, another word, which it shows that vicariance is something, some species are continuously distributed. Okay, in a landmass, and some geographical barrier will appear. Okay, so geographical barrier will appear and separate those populations. Suppose pit vipers are distributed in Sri Lanka, India, and Sri Lanka and India was a single landmass, and so the sea was formed, and the Sri Lanka and India got separated, and the new species formed in two different areas, Sri Lanka as well as Western Ghats. So they become a different species, species A and species B from a species A only. Okay, so that is called vicariance. So to know this, what is happening in the vine snake and what is happening in the pit vipers, I use these three methods to calculate their historical biography. So let's go to the result and see what are there. Come to next slide. Sorry. So let's see the historic biography of vine snakes here. Okay. So this is the analysis and results showing here clearly in this place. You can see the legends in the top: Sundaland, Asian mainland, Sundaland. All the colors in the color boxes indicate the region where the species evolved actually. Okay, and the pies, the round circle and pies are showing the different analysis on each clade of the branch. So basically, it says that some of the vine snakes actually originated and diversified in Western Ghats. Okay, they are not from they did not come only from Malaysian region, but they also evolved in the Western Ghats itself. But the ancestor itself evolved in Malaysian region. Malaysian regions means Sundaland and all the areas. So you can see the uh, arrow where again it says that there is a orange color arrow. It shows that the ancestor actually evolved in Western Ghats. So pine snakes are actually evolved in Western Ghats, and there are some back dispersal happen. Dispersal means the species originated in Western Ghats and they are again colonized to the Southeast Asia, to the Indochina region, Indo-Malayan region, all the places they again colonized to that region. That is called back dispersal. But also, it also gives some idea that some independent diversification and in situ radiation happened. Means diversification process, formation of species, and number of formation of radiation. Species started moving from the place and started changing the morphology and everything. They started from the Western Ghats. So Western Ghats is one of the important part for the species radiation in the snakes, and particularly for the vine snakes. Okay, go to next slide. And also it provides the time period. You can see the time period when it says that 25, 26 million years back they got separated, and that the diversification process actually happened in 26 million years back. Okay, next slide, please. So this is the historic geography of pit vipers. So how the pit vipers evolved, how, where, and how they diversified. Okay, so this is the, if you look at the tree itself, and you can find two boxes. One is red color box in the top. One is a green color box in the below. So this is a dash box. Okay. So basically, and in the bottom line, you can see the million of years when these species are diversified from each other. Okay, and they separate from each other. So that is what molecular dating does. This is a chronogram tree, and that is what molecular dating does. So basically, what we found, there are two species radiation happening in this in pit vipers. One happened in the Western Ghats in peninsular India, and one happened in Southeast Asia. Southeast Asia means Udala and Asian land. Basically, all the parts of Southeast Asia, Thailand, Myanmar, Manipur, all the including Assam also, Assam and Eastern Himalaya, Arunachal Pradesh, all the places, including South China, including Taiwan, and most of the places like the Philippines and Borneo, all the places are called Southeast Asian places. So in that region, one diversification process happened, and those species are also present in India, but they are present in the central India. They are not coming to the Western Ghats. Okay, but one of the clade group who are sister to those clade, the South Asian clade, they are our Western Ghats species. Okay, they are just evolved in the Western Ghats. The down box, the green box you can see, they are evolved in the Western Ghats. So the orange colored dots, red colored dots, three dots are there in near the red box. It's showing that the diversification, speciation, in situ radiation, everything happened in the Western Ghats for the primates separately. And those species from the Western Ghats species, they started colonizing to the central India. So one of the species I was talking about, the bamboo pit viper, which is found in Odisha also, that is actually originated from the Western Ghats and found in Odisha. Okay, so in central India, two species are present. One is bamboo pit viper. Three species, sorry. One is bamboo pit viper. Two species are from the northeast section. One is the Erythroros, that is Erythroros pit viper, and another species is Salazar. Recently described, somebody described this species. So those two species, they belong to the northeastern section, and this one species belongs to the western section, and they are meeting each other in the central India from a different lineage. Okay, so three species are in the central India, and more species in the western Ghats, and more species in the southeast Asian region. Okay, next slide, please. So one of the things I did in this study, that to measuring the rate of speciation diversification and speciation happen. So this is a process. This is a measuring process. How you can measure whether the speciation is happening right now, whether the speciation is diversification process is happening right now, whether the in situ radiation is happening, and so this is one of the process method where you can measure them. So this method is called the LTT. So a lineage through time. So how many lineages formed through the time? Suppose for last 30 million years back, how to now, how many species are formed? So what we found in the left side, if you look at the top plot, this entity plot shows that each step has a new species. So each step shows you the new different species. So the steps are going continuous to the top level till today, till zero date, means till today. So there are more species. There are like 20 species formed within last 30 million years back. Okay. So in case of pit viper in right side, if you look and there are something 20 plus species which are formed in the last 30 million years back. But what happened when you compare these two plots, you can see the right plot is little bit slow. Right plot means right side pit viper plot is a little bit slow in speciation event because pit vipers are very slow. They don't their habit and habitat is different. They don't move much. They don't colonize to different areas quickly. And but the vine snakes are faster. They are going. They can colonize and they can explore the new habitat. They can explore the new niche. So they become the different species very quickly. So that also can be measured from a plot. You can see the down plot of this bottom plot, right side one, that this histogram where the arrow indicates that vine snakes actually positively specific, means this specific event is still occurring. They are speciating from each other continuously. Okay, but in case of the I have not shown the plot for the pit vipers. In case of pit viper, it is negative. Means pit viper is stable. They are not specific, but vine snakes are non-stable. They are constantly specific. This diversification in situ radiation is constantly happening in vine snakes basically. So another plot, if you look at the comparison between two snakes in the left side, the black color plot where the snake images are there. So if you look at the images of the Asian pit viper and vine snake, the Asian pit viper took a slow gap and become slower in the oriental region. Okay. When you look at the vine snakes, it is like they are very fast. They are very fast and going on. So there is a comparison between both vine snake and the pit viper, both in the oriental region as well as in western Ghat region. Okay. Go to next slide. Next. So this is the end of my research work. So I continuously carry out, I understand the evolutionary processes and all the snakes. So this is one of the group. I have not worked on them. Probably I will work on them. This species is called the homegrown pit viper, which is found everywhere in the Western Ghats. Okay. So it is a lowland species. It is not a highland species. It never goes to the rainforest areas probably. I have seen them in the down of rainforest around 800 meters and all. From the 100 meter, all the grasslands, all the leaf litter they like. They are the ground dwelling pit vipers. They like the leaf litters, and they are venomous pit vipers. So this pit viper, there are three species found in South India. Basically in Sri Lanka, and two species are there. So one species is Hypnali, which is present in Western Ghats itself. Okay. So everywhere it is present, that Hypnali, the down one. But Hypnali Nepa and Hypnali Jara, they are present in the western in Sri Lanka. But they have a sister which is called Malayan ground pit viper, Calocelasma, which is also similar looking morphological and everything, and it is found in Malaysia. So this species, this Hypnalis, they are found in basically found in Western Ghat and Sri Lanka, but that is Malaysia. How? There is a large land gap in their distribution. So you don't get these species in the central India or maybe not even in the northeast India, but they are found in the Malayan region. This is one of the questions, how they got separated. Okay. Next, go to next slide. So this is another group of colubrid snakes called cat-eye snakes. Odisha has almost 3 species present in Odisha, 4 species present in Odisha. So there are more endemic species found in the Western Ghats, and also I'm trying to understand and how they are distributed, how the diversification process happened in the Western Ghats section. This is one of the group I'm working, and my work is currently in progress. Okay, go to next slide. So next. So this is the end of my topic, where I almost summarized about the how the snakes, how the endemic snakes are existed in the Western Ghats, and how they are distributed in peninsular India, and how they are distributed in the Western Ghats itself. And I also introduced about how the current taxonomy works. Now the current taxonomy is very interesting rather than the very traditional old taxonomy. But I always value the old taxonomy because it is one of the main base is provided to the current taxonomy. So mostly now people are using the integrated taxonomy. Integrated taxonomy is whatever I explained in the taxonomic section, people use the taxonomy to derive the species and separate the species from sister species. So they make the new discover the new species on the integrated way using genetic data, geographical information, and all things. Also, I described how the diversification of in situ species relation happened in the Western Ghats. Okay, go to next slide. So this is the end of my topic. Uh, I would like to acknowledge some of the people connected to some of the places and funding agencies who gave me fund for this work. So basically, I like to acknowledge my professor, Dr. Karthik, who is my PhD guide, and I have a collaborator from UK, United Kingdom, Anita Malhotra. I work with her for the pit fiber and all the snake species. And also SK Datta sir, who was a retired professor from North Asia University and Utkal University, and he was he's one of the renowned herpetologists in India. He's the best. He's the one of the major inspiration to my life, and because of him, I'm in this field today. Okay, and so I would like to acknowledge my colleagues, lab colleagues, and collaborators. Lots of collaborators I have from all over the world. Currently, I'm doing lots of work with many collaborators from UK, US, and USA and Iran and all the places, Europe, German countries, French countries, and all, even Sri Lanka and all the places. Uh, so they are the great help values for me. Also, I like to acknowledge the field staffs when I'm doing field work in the tiger habitat, in the elephant habitat. My field staffs are one of the major people who saved me from every situation and all. And so I got permit to collect sample from the many places in all over India. Okay, so I took permission from the almost 11 states from India. I think I am one of the PhD student who got 11 state permit from all over India. Okay, so there are like lots of funding agencies who gave funding to IIC, my supervisor, as well as some gave funding to me directly. Uh, my supervisor who got the funding from the Department of Biotechnology, IIC gave me money for traveling and field work and scholarship as well as as a PhD student. And critical ecosystem and partnership fund they gave a huge grant to my lab for the understanding the herpetologic fauna diversification process in the Western Ghats and Peninsular India. Also, Ministry of Environment Forest, they keep supporting to my department there. And from European Commission, I got money for traveling and working in UK and all the places. Thank you all for your attention, kind attention, and listening to my topic. So, would you like to ask some question? Do you have any queries? Ask anything?
Okay. Really, so so grateful to you. Really, it's a very nice session. Lots of information and useful one. I think all the participants would have enjoyed it. I can't find words to describe to thank you, sir. Really, it's amazing and a very nice session. Now I would like to request Sushriman, please, if you have any questions, if any participant has.
Okay, I will
Okay,
ask.
okay, sir.
I have collected some questions.
Okay, sir.
Sure. I have collected several numbers of questions from the participants
Okay,
and
okay,
also
sir.
from the co-convener of my department. So, Stephen Lidapati also. So, first question is, sir, in one of the your slides, you analyzed five genes from mitochondrial gene and a nuclear
Okay, sir.
gene. So
Okay,
why
sir.
you chose those five particular genes? Is there any
So, sir,
significant?
actually, what happens in biographic stories? Biographic stories, a which gene is needed basically to get the depth, the divergence depth, basically. So, mitochondrial genes are very, very fast evolving genes. They evolve years. They are not conservative genes. They are not much of conserved. So they are like they evolve very quickly. They evolve for, within a million, within a 1 million year also, within a 50,000 years also. They evolve very, very quickly. So we use three mitochondrial genes to see that because they are and so that can give you the species of event and give the morphological structuring of the species. Also, we use the nuclear genes to get the depth, basically the divergence depth. So it will give you 50 to 300 million years of gap in the molecular dating, molecular clock. So in biographic stories, molecular clock is a major sole part, is a major part which is always needed, which is being required. So you need to understand and calibrate the molecular clock so good, so robustly, so that you can understand the biography of two regions, particularly how the species are distributed. You need to understand. Then you need to use the nuclear gene. Nuclear gene provides more data. Nowadays people are using more nuclear genes, more. Some people are using 100 nuclear genes because of the next generation sequencing. It is possible to use lots of nuclear genes. So people are using lots of nuclear genes nowadays in the biographic
Okay, sir.
stories. Yeah.
Okay, thank you. So another
Yes, sir.
question from Dr. Abhimanyu. He asked
Okay, sir.
what factors influence species radiation? Are
Species,
endemics?
species,
Huh. Okay, sir.
species radiation. Species radiation means what happened was there are lots of factors. There are lots of historic factors, ecological factors. Lots of factors are there. One of the factor is the competition between the species. When there are two species are there, competition happen for the research. Suppose for food. Suppose simple as food. Particular single species plant. Suppose some two butterflies are eating a single plant, then they are competing for that plant. Then one butterfly will select and go compete for go and select another plant. So plant B. So the plant B, the different species plant, it has to evolve according to digest that plant. Okay. So the predation process happened there itself. So like that, when there are like many factors like competition, like predation, like environmental factor. Also, there is a species radiation because of the climatic factor. Glaciation system, glaciation process. When the glaciation happens, what in every 10 to 20,000 year gap. So it separates species, particularly single species. It will separate. Okay, separate by cutting them their genetic isolation. They geographical they will be cut off. They cannot meet. So they become different species. They evolve as different species. So
Oh,
radiation
okay,
can
sir.
be radiation can happen from any source. Radiation can happen from any source.
Okay, thank you, sir. Another
Okay,
question.
sir.
Are endemic species to the Western Ghats the product of species radiation?
Yes, they are species radiation because most of the species event happened with a small area because Western Ghat is a humid forest. During climate change, sorry, not climate change, it's a glaciation event. Always the lower India, the peninsular India is covered with ice, but the Western Ghat section never covered with ice. So most space provides them expansion and reduction of the space throughout the for last million of years. The species got separated again, mixed species got separated, and the computation and everything, all the factors are influencing them so much that they got separated and they got diverted and they started radiation. So when the ice and everything melt down, so some species become so specialized, so they wanted to go to the explore to the new habitat. So they got separated and they become different species in the lowland and everywhere. So they went to the lower parts and lower habitat and they become separate species. So species radiation is happened from the Western Ghats technically. You can see I explained one species is already colonized from Western Ghat to the Southeast Asia. Means that species are already radiated from Southeast Asia in my study. So
Okay,
there
thank you,
are,
sir.
yeah.
Can you exemplify few evolutionary trend happened in this biodiversity hotspots area?
This is evolutionary trend means what sir? What I didn't get the again explain.
Can you exemplify few evolutionary trend happened in this biodiversity hotspot area?
Yeah, this is what species radiation basically is diversification species radiation. And not only snakes, there are many snakes, there are many frogs, there are many other mammals, lots of fishes are there, which already evolved from this region. The species radiation and diversification process already happened for the many of the species because of the all the past historic and biogeographic time and all the historic time historically the ecology and everything influence them to be different species. So this the mostly the trend means I don't know what he actually exactly means trend. I don't understand, but the species and event happened for many species actually.
Okay, thank you.
Yeah.
Then what is cryptic species? What is the
Cryptic.
difference between cryptic and sibling species?
So cryptic species. Sorry, sibling species.
Sibling species and cryptic
Okay.
species.
Suppose sibling species are basically sister species. They are the sibling species. Means species when you easily I can say easily I can give you example of a sibling species. This vine snake, suppose that vine snake I told you about the Western Ghat vine snake, two species present in two hill ranges, species A and species B, dispar and paratoti, you can say. So dispar and paratoti are sibling species, sister species. They are sister to each other. You can also say them as sibling species. Okay, but cryptic species is something you cannot identify that species. Suppose in a population, when you look at a population widely distributed, single species, you always think that there is a single species. But when you do the genetic data analysis, you can find the genetic information and you can see the species are actually separated internally. They are genetically already separated. So that is a cryptic species. Means they are the morphology is similar, but genetically they are divergent. That is cryptic species.
Okay, sir,
Means
thank you.
yeah.
Another question from Aryajit Satpathy from BJP Autonomous College, Bhubaneswar.
Okay, sir.
When we say Western Ghats is biologically more rich than Eastern Ghats, is it because Western Ghat is more explored and researched?
No, it is not true actually because Western Ghat, Eastern Ghat is Eastern Ghat also explored. That Eastern Ghat has very few species to be found. Okay, so Eastern Ghat actually in case of snakes and all the things, Western Ghat is habitat itself is different because Eastern Ghat has no rainforest habitat. But Western Ghat is completely rainforest habitat, and always rainforest habitats always give the species diversification process high. So always the species business. Look at the Amazon, look at the Borneo, look at Sri Lanka, look at all the hills, all the regions in Southeast Asia. Those are all hill rainforest habitat. Rainforest means very always rain is there, and the habitat is very humid and trees are very the evergreen trees are there, so it provides more opportunities spaces on event. So species there diversified there. So as compared to the Eastern Ghats, the Western Ghats is like that, but in the Eastern Ghats you have only dry deciduous forest everywhere. You have scrubland. In Eastern Ghats you have mostly in the southern Eastern Ghats you can get little bit moist deciduous forest, little bit moist deciduous forest. And the difference between deciduous and rainforest is that deciduous forest they shed the leaf every year once. So during summer they shed the leaf, but the rainforest they never shed the leaf. So basically in the rainforest when they never shed the leaf, that every rainforest tree has a every leaf has a lifelong lifespan of 15 years or 16 years. So 16 that 16 years of time provides that tree to so much cover the habitat will be so much better for species to speciate and food and everything will be so available, the speciation even happen. But in the Eastern Ghat, we have only dry habitat and mostly scrub forest, as well as with the terrain is important. One of the terrains. So terrains are not supportive. So you don't get much of the Eastern Ghat section. You don't have water. You have to drier part. It is not humid at all. But Western Ghat it is always humid. Those are cloud forest. You always get rainfall there. After 3 o'clock, you get rainfall in the Western Ghat. Also, Western Ghat is like almost 8 months you get rainfall. 8 months you get rainfall. How much rainfall you get in Eastern Ghat? You don't get much of rainfall in the Eastern Ghat. It is not about the exploration. It is about the habitat itself. So the habitat is so much better that number of species in Western Ghat and species is more. But Eastern Ghat it does not have that kind of habitat. That's the reason. And the terrain is also important. The terrain and is very rough. Eastern Ghat terrain is not that much of rough. You can access everywhere. That's the reason.
Okay, thank you. In your presentation, you have told in Western Ghats, in some region, the temperature falls below 0 degrees centigrade. So,
Yes,
what
yes.
are the obstacles you faced during sampling and how you overcome those? Please share your personal experience.
Sir, kindly repeat that. I cannot hear properly.
Uh, in Western Ghat, in some region, the temperature falls below 0 degrees
Yeah,
centigrade. So, what
yeah.
are the obstacles you have faced during sampling and how you overcome those? Please share your personal experience.
Yeah, some part like Uti, most of the people visited the Uti because it's a high hill station in Nilgiri Hills. So from the Uti area, there is a field station. We have a personal field station, IIC field station in Uti, from 60 kilometers from Uti. That is called Mukurti Tiger Reserve, Mukurti National Park. So there the temperature is so low, it's almost like zero at night, and mostly we do the sampling in night mostly for the snakes, and daytime also we do. It does not matter. We faced many obstacles in some everywhere. Even I worked in Himalayas also. I worked in snowfall areas and all the areas. So it does not matter actually. So basically, we know for snakes in the herpetofauna when to sample. That is one of the important thing. You need to understand the behavior of the species to sample. Because I worked on snakes for last 18 years almost, 18, 17 years, 18 years almost. So I know snakes and their behavior and their habitat type. So wherever I go, I find the snake. That is the thing. So any habitat I enter, I can find the snake. So I am so expertise right now. I can find any snake anywhere. So I can go and select the select. I know the distribution. I go what time and what time of the year I have to go and find like that. So mostly in the rainfall season, always it is cold in the Western Ghats. The all that hilltops are cold, but still snakes are present there because the snakes are there like that habitat. It does not matter. We work lots, so it does not matter much because we know the habitat.
Okay, thank you, sir. And
Yeah,
another last question for you.
yeah.
Why pit vipers are not speciating?
So it is probably because of their behavior and food restriction. The kind of food they prefer, the resource they prefer, they are not getting that much of resource, and they are much of ecologically tolerant species. They don't many species from the Western Ghats. They are ecological tolerant species, except one species that bamboo pit viper. Many of the species they don't like to. They cannot survive outside the Western Ghats. That is the reason. So the precision level is low. So many in case of many pit vipers, I don't know about Southeast Asian region because those are rainforest areas. They are speciating, but slowly they are speciating. Yeah, slowly. As compared to the pine snakes, they are speciating slowly. If I compare with all the snakes, the speciation rate will be different for different snakes. So even in the monkeys, even in other animals, the speciation rate is different in different rate. So this is just a comparative analysis. I was showing that how the species and rate of diversification of process happen and all. So this is just they are specific. They are specific but slow as compared to the wild snakes.
Thank you, sir. Thank you for your presentation. and thank you for your critical work which we have in Western Ghats.
Thank you, sir.
Thank you, sir.
Now, I would, before winding up, I would request Susriman to present vote of thanks.
Am I audible to all?
Yes, yes.
Yes, ma'am.
Very good afternoon to one and all. Myself Susri Sangita Patti, lecturer in Zoology, Department of Zoology, Udala College, Udala. I deem it a great honor and privilege to propose the vote of thanks on behalf of Udala College and Department of Zoology. In the outset, I convey special gratitude to our Honorable Principal, Professor Gopal Chandra Behra sir, for giving us permission to organize this webinar and also for his constant encouragement. I extend my heartfelt thanks to our today's speaker, Honorable Dr. Ashok Kumar Mallik sir, for enlightening us with his knowledge and research experience. Sir, indeed this was a very informative discussion. Today's talk not only gave us deep insights to the topic, but also revealed many interesting facts. Once again, I personally thank you, sir, for sparing time from your busy schedule to grace the event. Thank you, sir. I also thank our head of the department, Dr. Prabhakar Sahu sir, whose effort and willingness has made this webinar successful. I mention my deepest sense of appreciation to Dr. Prafulla Kumar Panda sir, Coordinator IQAC, for all his support in every possible way. I am also thankful to Professor Satyavrata Mallik sir, Assistant Coordinator IQAC, for his cooperation. The Department of Zoology and IQAC is thankful to all the participants present here for accepting our invitation and extending their support throughout the session. Last but not the least, I appreciate and thank all the teaching and non-teaching staff members of the college for their support and cooperation behind the screen. Once again, I thank each and everyone for their support and cooperation in making today's event a resounding success. Thank you very much for your kind attention. Thank you all.
Thank you, Susri.
One announcement here, sir. Actually, feedback link I have sent both in YouTube as well as in Google Meet here, but some issues are there in YouTube. It's not responding. So kindly, I request all participants, don't worry, I will send it to your personal mail. So kindly bear with us, and definitely you will get your certificates, and you have to wait for 5-6 days to complete the process. Don't worry, almost all of you will get the certificate. Thank you. Thank you, sir.